Good afternoon, colleagues, and welcome to this Usher event, which is a seminar series which we started earlier this year, New Frontiers in Health and Social Care. And thank you all for those who were able to brave the Scottish weather and make it here in person. And thank you all for joining us, those who are joining us online. So as you all can guess, this is a hybrid event, and we have colleagues, a lot of colleagues in fact, joining us online. And we'll have about 25 minutes of presentation, followed by about 15 minutes for our questions and answers. So if you are going to ask a question, please go onto the website to ask your question. Colleagues who are joining in person, the QR code is on the seats next to you. So you can scan the QR code and get online, and then onto the event website, and then ask your question. There is no planned fire evacuation, so if the fire alarm does go off, we all need to exit to the nearest exit in an orderly manner. But hopefully that shouldn't happen. It's important to also note that if at any point of time there is any other alarm, fire alarm going off, you should be mindful of that as well. So with that, I'd like to welcome the presenter, the keynote speaker for today, Professor Melita Gordon. So Melita is an NIHR research professor and clinical academic. She's accredited both in internal medicine as well as in gastroenterology, and she's got a career-long commitment to building research capacity and addressing major health challenges through equitable clinical research, partnership, and community engagement in Africa. Melita trained as in undergraduate medicine in both in Cambridge and Oxford, and then did her general medical training in Oxford, Zambia, and Belfast, and then did her specialty training in Sheffield, Malawi, Liverpool. So she's been all over the place. And she's focused her research for over 25 years on vaccine-preventable invasive salmonella disease, both typhoidal as well as non-typhoidal salmonella in Malawi, and she leads a clinical translation multidisciplinary program that encompasses epidemiology, clinical pathogenesis, pathogenomics, and vaccine clinical trials. She's received research fellowships from Wellcome, UK Clinical Research Collaboration, National Institute of Health and Care Research. She was also awarded the Sir Francis Aylesbury-Jones Research Medal by the British Society of Gastroenterology, and she serves as an invited expert on salmonella vaccines for both the WHO and Gavi, the Vaccine Alliance. And we are very pleased to have with us Melita, and the floor is yours.
Thank you very much. I'm thrilled to be here. I'm very honoured to be part of your seminar series, so thank you for inviting me. So, as you say, I'm speaking of really, I thought I would tell you an overview story about over 25 years, what it has taken to bring two pathogens through to the point where vaccines are being brought through clinical development and into use. So that's what we're going to talk about today. And we'll have I'm going to go fairly high speed and fairly helicopter view, and then in the questions you can focus on what interests you. So just for background here, there are two kinds of salmonella that cause invasive disease. There's typhoid, which is a classical tropical disease that we've all known about for generations, and then more recently emerged, we have what we call, rather unimaginatively, non-typhoid salmonella, which is all the others. And these, of course, are not typically host-restricted, but in the right host will cause very, very serious invasive disease with a high case fatality, and they're associated with HIV, malnutrition, malaria, and very young age. They mainly affect Africa, whereas typhoid is Africa and Asia. These are united by the fact that they have been driven in recent years by the emergence of antimicrobial resistance and by poverty and inequity. And the other unifying feature is that they cause enormous case fatality, huge number of deaths, and that they are vaccine preventable. I've given up being subtle about this. If you equate the number of deaths to the number of jumbo jet crashes, Deaths from typhoid globally are 32 crashes a month, that's 1 every day, and NTS is a crash every other day. Most of these are in children, certainly for NTS, most are in Africa, and most are preventable. So how do we bring them to a vaccine? I put a kind of schematic here of what it takes to get a pathogen through to being tackled with a vaccine. And it starts with surveillance and epidemiology, understanding the pathogen, understanding the host. Increasingly, CHIM is an important tool, and this has been the case for both salmonellas, then through to clinical trials, introductions, understanding how this is received by society and by the community, evaluations in real life, policy, and then modifying your schedules and boosters. And so we're going to take a view of that. The story I'm going to tell encompasses pretty much all of these, so it's going to be high level. And I'm going to tell it in chronological order for NTS, which I've been working on since 1997, and typhoid since 2012. So, the story of NTS started, I went to Malawi, we took blood cultures. The first pathogen we grew was a non-typhoidal salmonella. I was a bit mystified, and it became clear it was the commonest. I phoned all the world experts that I could find, very big phone bill from Malawi, said what's going on and why is this happening. They all said, didn't even know it was a thing, haven't got an idea. And I thought, oh, I seem to have found a research area. And we quickly found that it was common in adults and children, non-typhoidal salmonella disease, and it was bimodal, with a peak in children and then a peak in adults that was 97% associated with HIV. And the second thing that was very evident was that this was being driven by in epidemics through the emergence of antimicrobial resistance, and we were able to sequence. This was back in the days when sequencing one pathogen was like a huge deal, and we found that there was a rather distinctive Salmonella typhimurium that was host adapted in the African setting called ST313 clade, which underpins a lot of the invasive disease, and other strains have also become host adapted in Africa. So we have host factors and pathogen factors that have been playing a role. The striking thing about NTS was that it actually, clinically speaking, had a lot of commonalities with typhoid. So this was not just food poisoning overspilling into a little bit of bloodstream infection. This was persistent, high fever, recurrence, big liver and spleen, clearly a nasty, deep-seated systemic infection. We saw pneumonia, diarrhea, actually relatively uncommon. This is not a gut disease, it's an invasive systemic disease with a very high case fatality. So I like to have this little picture just to kind of say, in the community, if someone's exposed to a non-typhoidal salmonella in Africa, they might develop diarrheal disease, as you might in Europe, but if they are susceptible, they're more likely to develop this invasive syndrome, which is not simply an overspill from diarrhea. It's a deep-seated typhoid-like infection, and this is what we see in immune-compromised communities in Africa. Sorry, I got forward twice there. And this is really just to highlight when I first started, I recruited a cohort of 100 people. By the end of following them up for 2 years, which is what I did, 80% of them were dead, and those that survived, half of them had recurrence after recurrence after recurrence. We treat with antibiotics and would come back. So serious, persistent disease. And when we looked at the recurrent strains, each different colour here is a single individual. And what you can see is they were having a recurrence with the same phylogenetic type over and over again. So these were not just being reinfected over and over again, it was hiding away and recrudescing in their bloodstream. Similar to the kind of thing we see with typhoid. And so I kind of asked the question, can I prove that this is hiding intracellularly? And took blood and bone marrow samples and then used solid phase core plates to look at lysed and unlysed samples, and found that when we burst the cells, we had an increased yield. And this was the kind of primary proof, if you like, that this was an intracellular pathogen that was going into hiding and then recrudescing back out into the blood. So kind of primary evidence, really, of what was the underpinning pathogenesis of this disease. This is really important because you need to know how the disease works to tackle it with a vaccine. So I then spent longer than I care to think about, trying to unravel what were the immune defects that we saw in adults with HIV and in children. This is a kind of summary slide, but there were multiple defects. They were slightly different in adults and kids. We found mucosal defects, impaired innate cells, depletion of IL-17 cells. At the cellular level, we found a kind of collapse of the NF-κB-driven inflammatory environment in HIV, which impaired the ability of cells to deal with intracellular infection and this persistence. And then at the humoral level, we found these contrasting findings that in adults with HIV, they actually have too many anti-LPS, NTS antibodies, which create a kind of steric hindrance and prevent killing. Whereas in children, it appears that there is a defect and a lack of antibody response, which may underpin that. We're going to come back to antibodies in children a little bit later. And the reason that we're doing that is if you look at the process of developing robust protection, you can think of this as very much starting with the physical barrier of the gut, working its way through to the innate cells who don't have memory, they're the first line of defense, monocytes, macrophages, then through to CD4 cells developing memory and the adaptive immune system, and CD4 helper cells then support the development of cytotoxic CD8 cells, B cells, and the production of antibody, which is necessary both for cell-free killing and which supports intracellular killing. So if you look at it with this model, antibody really is sort of like peak immunity. It's probably our best marker of mature immunity, which is why we then went on and focused in on that. Looking at the age incidence over time in Malawi, each box is 1 year from 2010 through to 2019, over 10 years. And if you look at the age profile, this goes from young children on the left through to adults on the right. What you'll see is back in 2011, 2012, 2013, we had significant peaks in adults with HIV as well as in young children. But as the ART rollout across Africa has kind of taken hold and taken root, you'll see that those peaks in the adults have gradually declined over time, whereas among kids, we see just as much as ever related to malaria and malnutrition. So in terms of vaccine strategy, we're really focusing in on the kids as being the most important, significant burden of disease these days. And so if we then zoom in on what's the age incidence pattern, epidemiologists, this tells us a lot, or we hope it tells us a lot. And what we see is that there's relatively little disease early in life. We'll come back to why that might be. It peaks very young at the age of about 15 months, and then fairly rapidly falls off. So by the time kids are 3, 4, 5 years old, the disease incidence has fallen. Now that could be several reasons. It could be that they're not being exposed anymore. It could be that they are no longer subject to conditions that make them susceptible, or it could be that they're developing very effective natural immunity. Natural immunity from typhoid is terrible. You can have several episodes of typhoid and still be susceptible, but my hunch was that actually children were able to develop very effective natural immunity against NTS. That causes the decline with age, and that gives us a big clue for how we can develop an effective vaccine. So that's been the sort of important approach that we've taken. So again, if you think about how this might work, we know that kids in Africa are multiply exposed in their gut to salmonella and they have no symptoms. It passes around families, lots of different serovirals, pass around families and around communities. They don't have symptoms, and the result of those multiple exposures is either that you develop natural protection, that's good, or that you tip over and you get nasty invasive disease with underlying case fatality, and that's bad. And so this balance is obviously really important. And if you have a susceptibility, malaria, young age, malnutrition, that pushes you away from natural protection over towards invasive disease. So that's the way that we have been thinking about this, and so we've drawn that in a fancier diagram, which basically says exactly the same things. This is our hypothetical model. You have an enteric exposure, you develop cellular antibody, then you get immunity, then you go on sequentially to develop antibody immunity. This was demonstrated very nicely by Tenny Urenda, my local collaborator and previous student. The antibodies make use of complement peroxidization, and then this enables cell-free serum bactericidal activity and phagocytic killing and intracellular killing. And these stages are all affected by poverty, by sanitation, by rainfall, temperature, environmental issues, and also by host-specific susceptibilities like malaria, malnutrition, and anemia. So it's a relatively complex model. There's a lot to think about. And what we've done is set up a large community-based study in a malaria endemic area in the south of Malawi called Chikwawa, making use of a big mapped enumerated census and our knowledge of variable malaria transmission so that we know which areas are more at risk from malaria and not. And we've done a two-stage randomization and recruited 2,000 children spread throughout the year so that we even out seasonal effects. And we take 2 samples at day 1 and day 90 so we can see serological elevations. And we take blood and stool to be able to look at their antibody response, to be able to look at their stool exposures. We check their malaria status, check their anthropometry for malnutrition and weight. So we've accumulated now this large database. And in parallel with that, we also have serological samples from 3 other African sites so we can understand the variability from between sites, trying to interpret that in the light of what we know about the rest of their setting. So I said one reason my children might stop getting NTS as they get older is that they might not be exposed anymore or they might not be susceptible anymore, but I think that's clearly untrue. When we look at the stool positivity for NTS, and this includes the invasive kinds of serovars, we see that they are continuing to be exposed on the left there to NTS in their stool. So the exposures continue. It's not that they just don't encounter it anymore. And when we look on the right, we see very clearly this is RDT positive slides in asymptomatic healthy children. The pattern of febrile malaria disease is similar. So if you consider that children are getting the peak of NTS disease when they're 15 months, which is about the 4th column along, the susceptibility is still there. Okay, so all of this leads us to think that we are right, that the natural immunity that they're acquiring must be quite effective against disease. I've just put the age curve there so you can kind of appreciate the disease is falling even though they continue to be exposed and they continue to experience malaria episodes. So we looked at their antibody. And we found, encouragingly, roughly what we would have expected. In the very first few months of age, they have acquired antibody transplacentally from their mother. And this is IgG against one of our commonest strains, which is a Salmonella typhimurium. That wanes with age to a nadir at 6 months. That would be a pretty typical pattern. And then they start to develop their own natural immunity. We know from previous work that they've already developed cellular immunity, then they start to produce antibodies. So this is roughly what we expected, and then it plateaus out to a certain level. And if we overlay that with the disease pattern of incidence, we kind of say, is this a good fit? To me, it's not a perfect fit. You can see that disease goes up as the maternal antibody wanes, but they've really reached quite a high level of antibody before the disease incidence starts to turn around. So that suggests to me that it's not only necessarily about the amount of antibody or the amount of IgG. There may be sort of subtleties in the functionality, there may be other subclasses or isotypes involved in wearing protection. Cellular immunity may be playing its own role here as well in helping disease to be controlled. You may need some of those additional functions as well as just the amount of antibody before disease really starts to come down. Because there does seem to be a little bit of a time lag there. The other thing I would just kind of point out in terms of function, this is our antibody amount on the left. When we look at the serum bactericidal activity, we got a very big surprise here. We had expected that the maternal antibody would be producing bactericidal activity that was just as good as the antibody that children produce themselves. What we see here, this is total bactericidal activity, so it's not necessarily only what's provided by IgG, but the maternal antibody is not alone, is not cutting it in terms of cell-free killing of the bugs. It takes the children's own antibody to do that. So again, this to me is a kind of clue that says it's not as simple as IgG on its own in terms of protection. Okay, because the maternal antibody is possibly not as protective as we thought. Which leaves me with the question, why are young children not getting disease if the maternal antibody isn't, quotes, working? And I think the clue is perhaps, let's flip back here, is perhaps that actually they're not being exposed very much. Respiratory pathogens, if you think about it, kids breathe the air from the moment that they are born, right? GI pathogens, there's a lag. They're being breastfed, that carry around in their mum's back, they're not putting anything in their mouth even. So I think there's actually a bit of a lag, and I suspect that children are not being protected by their mother's antibodies as much as we thought, but more by breastfeeding, lack of mobility, and all those other things that protect a very young child. And they also don't have malaria to the same extent in that very young age. And just to give you a snapshot of the data that we are wrestling with, these are similar, very large serological data sets that we have with our partners in Burkina Faso, in Kenya and Ghana. And there are definite variability between sites. We've done here what we call a change point analysis, which is a model that allows us to see where the troughs and nadirs are. The bars at the top are the confidence intervals around the period of time, and you can see the level written in blue. And the thing that I find very interesting here is that Burkina Faso and Ghana on the right are probably lower disease incidence sites, but they actually have higher antibody levels. Whereas Kenya and Malawi on the left are high incidence sites, but we have low antibody levels. So to me that suggests that the antibodies that we're measuring are not actually a very good marker for invasive disease. They're probably a better marker for a susceptible population. This population has lower levels of antibody, they're more susceptible, and therefore they are a higher incidence. And that is actually fitting with some data from Kenya that was literally published yesterday, suggesting again that high incidence areas actually have lower amounts of salmonella detectable in the community, suggesting that they're not being exposed, they're not developing the antibodies, so when an invasive strain comes along, down they go. So it's counterintuitive to what we had expected when we started the project off, which is the story of my life, but it creates good research questions. So we did talk about the importance of functionality and the fact that the mere presence of this antibody doesn't necessarily seem to be enough to turn the tide. So we have recently started an additional Wellcome Trust funded project looking at the functional protection, not only the quantity but also the specificity of the antibodies and its functional qualities. In order to try and say, if we wanted one vaccine response, if we wanted one vaccine-derived antibody, that we wanted to cover all of these different susceptibilities that different children have, what would its characteristics be? What would it take for a vaccine to protect as many children as possible? That's the target. And so, as well as only quantifying the antibody, we're now just starting to move in, and Agnes Lockettzel, my PhD student in Malawi, is starting to work on this antigen-specific subclassing, affinity, and cellular functional high-throughput assays in collaboration with Billy Alter's lab in Harvard, looking at how the antibodies work together with complement neutrophils, monocytes, NK cells, and what's the functionality of this, and can we correlate the functionality to disease incidence as well as just the quantity. So that's where we got to with NTS. It has, I admit, taken me 25 years to get here, and we're only halfway up. But I'm also very excited to tell you that yesterday, for the first time, 2 volunteers were given NTS as part of the controlled human infection model in Imperial with Melita. So we're moving up this ladder gradually. It's a tough pathogen to work with because it's only very susceptible individuals, it's only in Africa, it's only young children, which makes this difficult, but we are chipping away. The second half I'm just going to talk with you about, I do have time, what's happened with typhoid, because that is a story that started later but has leapfrogged across MLW, across NTS, and we are now right through to national implementation, and this gives us a bit of a template for where we might go in future with NTS. So the story with typhoid was that in 2011, typhoid suddenly exploded into our blood cultures. In Malawi, we'd previously seen very little. You see it here in gold, and again, same story. It seems to have been driven by the arrival of multi-drug resistance. And in this case, we were able to look at the strains from global collection with the Sanger and Gordon Dugan, and to see that this was an imported strain that had come across from Asia and was now tracking across the continent. The little blue ones in the top right corner of the dendrogram are the Malawi's own strains, and we were able to dissect our own local transmission as well as across the continent. So, newcomer on the scene, And this caused an epidemic, and then we settled down to a stable endemic situation, whereas we are now. So we have endemic typhoid, which we never had before, because of the arrival of MDR strains from Asia. Lots of lessons about a global disease there. So we set about trying to find out what was the burden of disease in Malawi. We had a large strata consortium which spanned Asia and Africa. This is our fantastic field team. We did a census. We had 2 years of passive surveillance by blood culture for fever, accumulating over 600,000 person years of surveillance and follow-up, which makes me feel old and tired, because it was a lot of work. And then we looked at healthcare utilisation, serosurveillance, detection of carriers, to try and understand the burden and the transmission routes. What we found was that Malawi indeed had a very high incidence. You'll see that the peak of disease is somewhat older than it is for NTS with typhoid. And again, this relates to the fact that natural immunity from typhoid is not nearly as good as it appears to be from NTS. However, we were also able to ask the question, does this burden actually matter? And the answer clearly is that it did. It has a high economic cost to individual families. Remember here that people living in Malawi are living in one of the 10th poorest countries in the world. More than half the population are below the poverty line. So these costs are catastrophic for a family. So for every 100 typhoid cases, 44 families are bearing what we call a catastrophic financial burden, causing them hardship and dis-savings. 21 get serious complications, and that includes 4 will have perforations and 2 will result in death. So it's a serious economic and health burden. Unlike for MTS, we were in a happy situation that typhoid conjugate vaccines had been waiting in the wing for some time, and they came to fruition just as typhoid made this leap across to Africa. So we had a greatly expanded burden of disease globally, and we had vaccines nearly ready to go. And we were able to contribute all of our epidemiological data and evidence to WHO. They put out a recommendation for typhoid conjugate vaccines, which had been partly tested using controlled infection models in Oxford. WHO pre-qualified and recommended TCV for use in endemic areas and areas with high antimicrobial resistance. Gavi opened a funding window and recommended vaccines should be introduced by campaign, because it has a broad age distribution, so we should start with a campaign from the age of 9 months right through to 15, and then it gets introduced in young children in routine immunisation. So this was like the perfect scenario, really. Until COVID came along, this was one of the kind of fastest vaccine stories, from seeing the disease through to bringing it out. I mean, COVID has now dwarfed this achievement, but from the first case that we saw to rolling a vaccine out of Malawi was 10 years, which in old money is super fast, and would still be very fast for Africa. So we conducted a phase 3 efficacy trial that produced really the major data from Africa, alongside trials in Bangladesh and Nepal. We recruited 28,000 children with a primary endpoint of vaccine efficacy and secondary endpoint of tolerability and immunogenicity and safety. Vaccine was safe, it was highly immunogenic, and this is our trial recruitment. We had about 14,000 children in each arm. These are enormous numbers. We had 40,000 surveillance visits. We took more than 10,000 blood cultures, and we had a 1.3% rate of typhoid among the blood cultures. And to kind of cut a long analysis short, at 4 years of follow-up, we've been able to show that there's an overall vaccine efficacy of 78%. At 1.5 years, it was 80%, so it seems to have good durability of efficacy, and a number needed to vaccinate to prevent 1 case of typhoid of 163. I don't have time to show you all the cost effectiveness, health economics around this, but it works. It's very cost effective. These numbers make it very cost effective for African governments when we look at the financial cost to health services, the financial cost to individuals, the willingness to pay by governments to prevent a case of vaccine, and the cost of the vaccine at this rate. So it all stacks up economically for governments to roll this out, which was very exciting for us. These are the Kaplan-Meier surveillance curves over 4 years. The little flattening out in the middle is because this trial's found COVID. And we had lockdown in Malawi. Well, we had to shut the trial down for a short period in Malawi. But what you can see is that there's rapid separation of the arms, but they continue to separate over time, which suggests that the vaccine has ongoing efficacy. You still see a difference. So it seems to have durable protection, which matters for typhoid, because you're getting typhoid right through into young adulthood. And when we look at a meta-regression of efficacy year by year, across the duration of the trial, we see that the efficacy only falls by around 1% per annum by our best estimate, which again is great because this is not a disease like NTS, which has a narrow age band. This vaccine has to keep working for many years. So again, this was a really exciting output from the trial, and that's impressing the Lancet, so you'll be able to see the original data. In fact, it's in a preprint as well. The other important finding from this was that it didn't only work in the older age band of children, it worked right down. It was significantly protective, even in the very youngest age bands of children, because ultimately this is going to be introduced into routine introduction at the age of 9 months or 15 months, so we have to know that it works in the youngest children as well as in the school-age children. So very important outputs from the trial, which we were very happy about. So we're now at a position where we have 3 trials, our trial in Africa and 2 in Asia. These are really the only long-term TCD efficacy data that there are. It's effective, it lasts, it has good durability, and it's cost-effective. And this led the Malawi government to make a decision to roll it out. They applied to Gavi, we provided them with all the data that we could, and we had a successful application. Back in May, we had a national rollout and a huge campaign, and it's now in routine introduction in Malawi. So it's been a really historic year in Malawi. So this is the vaccine arriving in the country. A number of you may remember that we had Cyclone Freddy, which was terrible for Malawi, and that landed up right on top of the dates when the campaign was meant to be happening. We had to move it by a couple of weeks. We couldn't get the vaccine everywhere we needed to. These are the national EPI team literally wading the vaccine on their heads and the syringes to where it needed to go. They put it in a cart, they slept with the vaccine to keep it safe. So this was a huge undertaking. We were vaccinating everyone below 15, that's half the population of Malawi, vaccinated in the space of 10 days. It was an enormous undertaking, and the national EPI, we didn't do it, the national EPI team did this. It was a real landmark, and this is, you can see it was from young children right through to school age. So these are the campaign going in, targeting 9 million children. And so now our task is to say, did that work? So our continuing work now is we've done the trial. We're actually doing a preschool booster study to address this question of disease going on into young adulthood. But we're doing large-scale surveillance now. We did surveillance for 1 year before the rollout. We're continuing to do 2 years surveillance afterwards to say, it worked in the clinical trial. Does it actually work in real life? And the approach we've taken to this is we have adults and children kept under surveillance for fever. Anyone with a fever has a blood culture. That will include children who are in a vaccine-eligible age, some of whom will have been vaccinated in the campaign and some of whom will have not showed up and been missed, and adults who won't have been vaccinated because the campaign stopped at the age of 15. And then what we do is we can assess effectiveness by comparing the groups who are culture positive and culture negative in what's called a test negative design. So we look at those who don't have typhoid and those who do have typhoid, and we compare the probability that they were vaccinated. And the difference in those probabilities gives us what we think the real-life effectiveness of the vaccine is. So really nice study design. And then in terms of impact, we're looking at an ecological picture. What was the impact in children, given what we know the coverage is, compared to what are the patterns of disease we see in adults who definitely weren't vaccinated? And that gives an estimate of the real-life impact. So we have 2 measures from this ongoing surveillance. So to me, the best trick that we can do now is that we can use this experience that we've had with typhoid to now piggyback NTS on and see if we can get from where we are now with NTS, which is trying to understand immunity, using a CHIM, understanding what a vaccine response ought to look like that could protect these children and bring this through into policy. So over the last few years, we do now have, there are actually two vaccines. There's an LPS for German conjugate vaccine, which is in early clinical trials, and then there's this German membrane-based NTS vaccine, which is the one that we are working with in Malawi. It's being developed by GSK Vaccines for Global Health, a not-for-profit vaccine arm of GSK. These vaccines are the subject of a large consortium funded by the EU, Horizon 2020, very important funding, little plug there. And our part in this has been understanding cost of illness, understanding the burden of disease and awareness, and also the epidemiology to understand what will be protected and what's required at different sites. And I'm completely over the moon thrilled to tell you that this very week, we were, the team here in Malawi, this is the team on the top left, gave the first GEMA vaccines ever to anyone in Africa. So the first NTS vaccine has gone into the first person in Africa this week. So really, really, really exciting. This is the team literally standing outside the vaccine. It's all blinded, of course, it's clinical trial. They're standing outside the door while the first participant is randomised and having their vaccine in the room. A real sense for the team that they have made history in Africa. So this is what we call Salmonella vaccine. It's actually a triple-valent vaccine against typhimurium and enteritidis, which are the two commonest invasive serialized of NTS, and the typhoid conjugate vaccine, VI-CRM, which is actually the vaccine that's in the national campaign in Malawi. It's a phase 1b/2a clinical trial of immunogenicity and safety. It will run over the next year and alongside our partners with GBGH. There are other trials imminently going to start in Africa. There's another one in Kenya, and there'll be an age de-escalation trial going on in Burkina Faso a little later in the year once some of the data come through from this. So we're close to bringing this through now to a full loop and getting NTS into use in Africa, which excites me enormously. I just must give a shout out to the early career researchers and postdocs who have done a great deal of this work over the years, both for typhoid fever and NTS disease. James Mearing and Phil Ashton, bioinformatician, Esme Chirwa and Helen Dale, who worked on the Saints NTS study, Dari Jambu and Gaziwa, and Agnes, who worked on the immunology and the interplay with the microbiota, and the vaccine trials have been masterminded by Frank Patel, who is a force of nature, and Fumbani and Haggai both did some of the work on the cost of illness of typhoid and NTS, respectively. So, fantastic team who have really helped to bring both of these stories through. Of course, our national partners in the Ministry of Health and the local university are extremely important. We've benefited from being in some very large and very helpful consortia, including the Strata Consortium, the TIVAC Consortium, led by Andy Pollard and Kathy Nussel, respectively, and the iNTS Consortium, led by Jean-Luc Regi, with other international partners. And I particularly would like to point out the role in the immunology and the understanding of susceptibility that's been played by now Associate Professor Tony Nyirenda, who's a previous student of mine and now a long-time senior collaborator, who's really spearheaded a lot of the work on the seroepidemiology and laboratory methods in Malawi. Thank you to our funders, and above all, thank you to our study participants and the communities who have accepted this work and really embraced the progression through to vaccines through this. We absolutely can't do this without not only individuals but whole communities really buying in to what's happening. I do have an ulterior motive for being here, which is that MLW is just completing this fantastic new facility that's co-funded by the Wellcome Trust and also by Liverpool University and the Liverpool School of Tropical Medicine. And it is a new major clinical centre for clinical research excellence and to include open resource to training for Malawian scientists. So it's going to have a collaborative floor on the ground floor. It has free access 24-hour postgraduate library on the second floor. There's 2 new floors for research groups to house our expanding portfolio of research. It's going to double our footprint for research, and the top floor is going to be new advanced cutting edge libraries with advanced imaging, single cell sequencing and so on. And then you can't quite see it on the roof, we have party space. But you know, we need international partnership to do this. So really this is just a kind of call for collaboration. If you see stuff that we've discussed in this rather large sweeping story that you would like to discuss collaboration and training, please feel free. Either in questions or you can contact me by email and we can see if there are things that we can do. Thank you.
Thank you, Melita, for the wonderful talk. At the end, when you were describing the new building for Malawi, I thought you were probably describing our new building for the Usher Institute. And then when you said that there's going to be party space on the top floor, then I realized, no, that's not Edinburgh. That's probably somewhere where the weather is much more pleasant. But thank you for the wonderful talk. I think lots of take-home messages for us, particularly those of us who work in population health and public health, on how to take a vaccine from basic pathogen research to licensure and then to implementation. So I think we are on an exciting phase where we're going to see possibly in the next few years rollout of the vaccines for the non-typhoidal salmonella. So great work. Thank you. There's one question that's already come in, and they're asking the test negative design was used to compare the positive and negative cases, and whether you've looked at severe outcomes, e.g. hospitalizations.
Yeah, so we will be able to do that and look at severe outcomes. In the past, when we did surveillance for typhoid, we only did it at the tertiary referral hospital, and so the majority were hospitalised. What we found was that when we went out into the community and did blood culture at the secondary health facility, we were picking up like 5 times as much typhoid. So there's definitely a high amount of kind of subclinical mild disease that was previously totally missed. And we have to take account of that in all kinds of settings when we're looking at crude incidents from tertiary facilities and so on. So that's been an important lesson. But of course, severe presentations, hospital admissions, and particularly perforations, which are one of the most costly manifestations of typhoid, both in terms of human suffering, but also in terms of economic cost for the health service. So yes, we will be able to look at whether vaccination affects the overall burden, but whether it also is able to reduce that burden of serious disease as well, which of course is very important from several perspectives. I think the thing that is so good about the test negative design is that it is not biased by health-seeking behaviour. So it is a study design that should allow us to pick that out quite nicely.
Right. So I have a question, and I think, when you were sort of describing the non-typhoidal salmonella, I thought you were describing how this pathogen goes into hiding. And it sort of reminded me of tuberculosis, because it's one of the other pathogens there, where the pathogen continues to live in the body, but the immune system is sort of able to deal with it. But when your immunity goes down, that's when it's reactivated. and then you get active TB. And I was wondering whether that's something like that is happening with non-typhoidal salmonella, and also for someone like me who's not been exposed to non-typhoidal salmonella, if I were to go to Africa, what is the likelihood of any one of us who's living in Scotland, not exposed, goes and gets infected? Because although we are older people, but we're still quite likely never been exposed to this.
So that's kind of two questions. I'll take the first. The first one was about intracellular persistence, and I think, you know, we were able to definitively prove, if you like, that this is the case in people with advanced HIV. That, as I showed, is becoming less important in public health terms as the ART rollout has become a reality. Although ARTs do not completely remove the risk of NTS for people with HIV. They reduce it, but they don't remove it. Nick Feasey did some very nice work to demonstrate that. We don't want to go around taking bone marrows in children. It's very invasive. So we haven't been able to generate the same kind of prima facie event evidence in children of intracellular persistence. And clinically speaking, children don't seem to have recurrence in quite the same way. So it's possible that we know that the immunological risk factors in HIV and in children with malaria and malnutrition are actually slightly distinct. So it may be that this intracellular persistence is a more pronounced phenotype in people with HIV compared to children. So I think there's something of an open question there in children about the relative importance of intracellular and extracellular persistence, and therefore the importance of cell-free killing and cellular killing as well. And that's one of the things that we hope to tease out with this functional serology study, is to try and understand that a little better. So it's a very active question you put your finger on there. The second question was what?
With regard to someone who has not been exposed.
That is a fascinating question, because in what we're seeing is causing this disease in Africa are these serovars that have become slightly host specific. So there's a thing with salmonella that some are generalists and some have become very host specific, and the host specific ones typically cause really nasty invasive disease. And what the ones that cause invasive disease have done is they've ditched all the genes that they need to survive in the gut lumen and just kind of hang out in the gut, and they go straight for invasive disease. And you can see that footprint. It's called pseudogenization, and you can see that footprint. And what we saw with the NTS in Africa is that strains that would previously be diarrheal, they would be generalist strains that could infect many different species, have started to do this thing. They ditch the enteric lifestyle genes, and they look more invasive. They look slightly more like host-adapted typhoid, even though it's, you know, a salmonella typhimurium, which is a generalist. And so it's an absolutely fascinating question. To what extent do you need a special bug, and to what extent do you need a susceptible host? Because both of those components come together to cause this invasive disease. So I don't think you're that elderly, Harish. But the thing that this is in fact the first question that the CHIM is going to help address, because we don't really, we don't have the invasive strains here in the UK, so it's very difficult to address that question. So the first thing that the CHIM is doing is taking healthy adults in the UK, screened to the eyeballs to make sure that they're not going to get serious recurrent disease or gallbladder infection or anything else. And half of them will be given ordinary old diarrheal salmonella typhimurium, and the other half will be given African ST313 strains. And we hope that we will get a readout to see, are these strains able to be intrinsically more invasive and less diarrheogenic in a healthy host compared to an ordinary diarrheal strain. So we're very much hoping that we're going to start understanding the difference between those strains, and that will go a little bit towards addressing your question of what if you're a healthy person and you go to Africa and you're exposed to these African invasive strains. My hunch is that you probably need a degree of host susceptibility, as you referred to, as well as the invasive strains. I think you need both those components to really get a full invasive picture. But it will be very interesting to see what's the diarrheal readout and the invasive readout from the controlled human infection model. And like I say, I literally got a very exciting text from Malik Gibani, who is the PI leading the controlled human infection model in Imperial, yesterday saying, first 2 participants done, so we're on our way to an answer for your question.
So thank you for that, and I think just a follow-up question on the children, when you know you talked about the difficulties with bone marrow sampling, and especially the ethics of this in children. But have you considered post-mortem bone marrow in children who might be dying from, not from salmonella, but from other causes? And so one of the options would be possibly collaborating with the CHAMPS network, for example.
Yes, yeah. No, I mean, we have, of course we have. There's obviously, there's always acceptability and ethical issues around limiting post-mortem work. I think the difficulty that we ran up against when we were considering that is that when we did the work in adults with HIV, we were able to take these samples at the front door before they had any antibiotics. Whereas children who will have succumbed to this will certainly have had multiple courses of antibiotics, very unlikely that many would be untreated. And I think that would limit the usefulness of that approach and probably not make it worthwhile the general kind of trauma of trying to set up a post-mortem study. So we chose instead to take this approach where we sample across the community, we look at children who we know to be susceptible and non-susceptible and try to understand the antibody and cellular functionality and understand it from a community-based angle rather than from a post-mortem angle. So that's the approach that we ended up clumping for. And give me 3 years, I'll be able to tell you whether it worked, but so far we do seem to be seeing differentiation of functional readout from antibody and cellular assays that vary between children with different susceptibilities. So I think we are going to have some useful readouts from that project in the end.
So we've got a question online. Natalia is asking, will pregnant women be a target group for the vaccination for non-typhoidal?
Lovely question. Very nice question. So you're talking about maternal vaccination, the idea being that it's a disease that occurs in quite young childhood, and so you might want to vaccinate the mother so that the mother passes protective antibodies down. So I think that's an attractive option in that the very early childhood vaccine schedule is extremely overcrowded. Even in Malawi now, which is a very pro-vaccine country, we are starting to see parents kind of go, what? Another vaccine? Surely, you know, not. It has really become a problem. Vaccines are great, but we do need to multiplex them, which is why we have our pan-salmonella vaccine to try and reduce the number of needles and so on. So vaccinating the mothers is a kind of attractive proposition to hit early childhood disease and reduce the vaccine burden on children. However, I did show you the data that shows that although children definitely acquire antibodies from their mother, in terms of bactericidal activity, there doesn't seem to be as much bactericidal activity as from the children's naturally acquired immunity. And we're really digging into the reasons for that. One reason could be that you have slightly different subclasses of IgG coming through, and you do have different subclasses come through the placentas, the ones that are naturally acquired. The other kind of very obvious one, perhaps, is that you don't get IgM transplacentally, whereas once children start being exposed, they develop their own IgM, and IgM is a very effective complement fixer and bug buster. So it may be that, you know, that gap is because they're not getting IgM from the mother. What we are not yet sure of is how much that matters. What are the actual protective correlates? What are really protecting these kids? Is it the IgM boosting their bactericidal killing? Is it the IgG that is able to cooperate with their immune cells and boost phagocytic and cellular killing. Until we know that, I think we can't really definitively say should we be going for the mothers or are we more effective to be vaccinating children themselves. It's a great question. I'm in a WHO consultation next week. It's one of the specific questions that they posed. And I think the evidence isn't yet there to advocate for maternal vaccination, but it may be that we can generate that.
Okay, another question. Is there concern about sequence type drift that could weaken vaccine efficacy, or is the vaccine durable against multiple non-typhoidal salmonella sequence types?
Okay, so again, very interesting question. We do have multiple serovirus causing invasive disease. For the last 10 years, there has been a very stable picture of two dominant serovars, which is Typhoid and enteritidis. The main thing that seems to have determined the serovar distribution is the presence of antimicrobial resistance. So you would hope that vaccine pressure might not affect that very much. However, there was one instance in DRC, in Congo, of an LPS negative strain emerging. And that, of course, is a concern. Anti-LPS antibodies are probably the dominant immune response. They're probably a major component of the response. So the fact that there could be either drift of the LPS molecule within the strain that you're concerned about, or that a completely different serovar strain could come through, is a concern. The different NTS strains are serovar specific, but protection is probably serovar specific. So if we deal with Typhoid and enteritidis, another serovar could emerge, it could develop antimicrobial resistance under the right kind of pressures. We certainly know that neonatally there are some strains, for example, Isangi, that cause very nasty MDR, nosocomial disease in neonates. Something like that could come through. So it is something that I think it's a question that really highlights the importance of ongoing proper microbiological surveillance after the rollout of vaccines and understanding the drift of bacterial populations over time.
Thank you. And the last question is from Alex. Is there an overlap with HIV in the population? And if so, what's the impact of maternal vaccine response in infants? Is there an overlap with HIV in the population?
Okay, yes. So in adults, NTS is almost exclusively associated with with HIV in the Africa setting. In the past, 97% of our adult cases were related to HIV. A small number would be the very elderly, a small number might be somebody with diabetes or malignancy, but HIV is the overwhelming association. As I showed in the slide, in terms of public health, that is declining in overall importance, speaking from a public health perspective, as the ART rollout really beds in and has been protected. In children, HIV in the past has been an associated risk factor, but again, as the mother-to-child prevention programs for antiretrovirals has taken hold, the number of HIV-infected children being born is now very small in Malawi, and that cohort of children who were previously not protected by maternal antiretrovirals is now passing through. So I think HIV, while it is important, while it is there, is relatively unimportant. Maybe that's putting it too strongly, but I don't think it's likely to be a major player when we come to think of vaccines in young children.
Thank you. And so we're coming to the end of time, and Melita, thank you so much for doing this. Wonderful talk. The seminar is supposed to finish right on time at 1:30 UK time. So thank you so much. Just a gentle reminder with regard to the Usher annual lectures, as well as inaugural lectures, as well as the follow-up in the new year, New Frontiers in Health. Do we have so we have an inaugural lecture for Usher professors That's going to be in about 2 weeks' time on Monday, 13th of November at 5 p.m. in the Playfair Library. Do attend if you can, and this is an in-person event only. And then we have the next event within the seminar series, which is going to be on the 25th of January, where Chelsea Moroney and Kathleen Metronoska are going to talk about integrating practice-based knowledge and research. So this is going to be quite interesting. Melita currently is an NIHR global health professor, and Chelsea recently was selected by NIHR for an NIHR global health professorship. So it'll be very interesting to hear from Chelsea's perspective, and Chelsea's been working primarily in Botswana. So do try and make that event if possible. And we'd also like to hear your thoughts regarding this particular seminar. So please go on the website and give your thoughts. And we'd like to continue having these events at regular intervals in the next year as well, and particularly once we move into the new building, sometime in spring, summer next year. We should be able to have more of in-person events over there. So the other important thing is that I'd like to make some words of thanks. So thank you, Melita, for coming over and doing this for us. Thank you to the Usher communications team for all the coordination and delivery of the event, and most importantly, a plug-in for Laura Marshall, who organized this event. Laura has now completed 19 years in the university. She's worked for 19 years. And many thanks, Laura, for all the hard work that you put in. And thank you all for, again, sort of braving the weather and making here in person. So thank you all, and thank you, Melita.
Thank you very much.